Unlock how CRISPR-Cas9 edits DNA. We'll explain its bacterial origins, the molecular mechanism of guide RNA and Cas9, real-world applications from agriculture to medicine, and ethical implications researchers face today. Chapter 1. Origins. CRISPR as a bacterial immune system. CRISPR-Cas systems were discovered as an adaptive immune system in bacteria and archaea, first identified in 1987 and characterized in detail by 2007. Clusters of short palindromic repeats, CRISPR, store snippets of viral DNA, enabling recognition of future infections. Cas proteins, notably Cas9 from Streptococcus pyogenes, use stored spacer sequences to target matching DNA. This natural molecular memory allows sequence-specific cleavage of invading genomes. Researchers repurposed the system in 2012 to cut any chosen DNA sequence, in vitro and in cells. Understanding this biological origin explains why CRISPR provides programmable precision and why guide RNA design and PAM sequences are crucial constraints. Chapter 2. Mechanism. How CRISPR-Cas9 finds and edits DNA. CRISPR-Cas9 editing uses two molecular parts: a 20-nucleotide guide RNA, gRNA, that base pairs to target DNA, and Cas9 nucleus, that introduces a double-strand break 3 nucleotides upstream of a protospacer-adjacent motif, PAM. Typically NGG, the break triggers cellular DNA repair. Non-homologous end joining, NHEJ, often creates small insertions or deletions, indels, and is efficient, up to print 70% editing in cultured cells. While homology-directed repair, HDR, can introduce precise changes but is rarer, often 1 to 20% without selection. Off-target cuts can occur at similar sequences. Engineered, high-fidelity variants and truncated guides reduce unintended edits substantially. Base editors convert C to T or A to G without breaks, often reaching 40 to 60% efficiency. Prime editors use a PEG RNA to write edits. Chapter 3. Applications. Concrete examples in agriculture, medicine, and diagnostics. CRISPR's practical impact spans agriculture, medicine, and diagnostics. In crops, researchers have produced disease-resistant or shelf-stable varieties. For example, gene-edited mushrooms and tomatoes with reduced browning passed regulatory review in some countries. In medicine, ex vivo editing of hematopoietic stem cells targets sickle cell disease and beta thalassemia. The experimental therapy CTX-001 produced sustained clinical benefit in early patients by reactivating fetal hemoglobin. In vivo approaches aim at retinal disease using AAV-delivered CRISPR and liver-targeted lipid nanoparticles. CRISPR-based diagnostics such as Sherlock and Detector detect viral RNA within an hour, demonstrating rapid, portable pathogen detection capabilities that aided research during outbreaks. Chapter 4. Advanced considerations, safety, delivery, and ethics. Advanced issues determine CRISPR's trajectory. Germline editing raises profound ethical and safety concerns because changes would be heritable, as highlighted by the 2018 case where embryos were edited, prompting global censure and moratoria. Somatic therapies avoid heredity but face delivery and immune barriers. Several studies found pre-existing antibodies or T-cell responses to common Cas9 proteins in roughly half of tested adults, risking inflammation. Gene drives use CRISPR to bias inheritance and could, in principle, suppress vector populations. Lab work reduced Anopheles numbers, but ecological risks demand rigorous containment and modeling. International regulatory frameworks, risk-benefit assessment, and equitable access will shape whether CRISPR benefits are realized responsibly. CRISPR-Cas9 transformed DNA manipulation by repurposing a bacterial immune system into a programmable cutter. Its precision, efficiency, and adaptability enable breakthroughs in agriculture, medicine, and diagnostics, but technical limits, delivery, and immune challenges, and serious ethical and ecological questions require careful regulation and public engagement.